Sorry about that. So more tweezers, very fine points. So this is the back tool that I was mentioning earlier. You see it, it's a dental pick. It's very flexible, meaning that as soon as you apply a little bit of pressure on it, it's going to bend. And then once the pressure is released, boom, it's going to work like a, yeah, it's back. So what I did is I took like an old screwdriver, basically, and I modified it myself. So I bent it and then I created like a very pointy end. And that is like very robust, okay? And now I can use basically this tool to just like apply a bit of pressure and then dislodge the calculus, it stays in place. So, you know, $35, free. Okay, I just did it myself. That's it. And that's my favorite tool. So we're not going to use that today. That's no need for that. Okay, and then now the Dremel tool. So here's the beast, the noisy beast. There's some of those tools are actually cordless. This one has a cord, so I need an adapter actually. Oh, yes. Oh. It's here. Okay. Where's the Aussie one? There.
Yeah. Maybe the other way around? Yeah.
No, I really think it's like this. Right socket. It looks like it is.
So,
yeah, that's why. Yeah, that's why it's
not the right.
Oh, that's for the output.
Yeah,
okay.
Output. Yeah.
Here we go. Hopefully it's not broken.
Just hit that. Yeah. Yeah.
Okay. And so you can use it like this, but you can see it's quite bulky. I mean, I don't like the control because it really is hard to be extremely precise with this. So usually what I do is I fit the extension cord, because this I can take it like a pen basically, and then I can work very precisely. So I prefer to use that. I'm going to simply put it in place. All these people watching me is making me very nervous. Okay, here we go. So now, cool. So I have my tool ready. And then in there, I have cutting discs. So basically just abrasive cutting discs that you can fit onto the Dremel. So when it rotates quickly, then it cuts bones. Of course, you need to be careful because this is, can break. It's quite fragile. So you need to be careful when you manipulate that, especially your eyes, okay? So this is why you have protective goggles. And then you have these bits that you're going to fit onto your Dremel tool. And basically, my size of choice is the 99. Do we see something? 9905. Yeah. So 9905 is really these Dremel bits where there's a round head that is the same size as the shaft of the bit. Okay. So not smaller, not bigger, just like the right size to do minimal damage to the bone sample and still be able to go relatively deep. And that's basically going on top of this. And here we go. Now I'm ready. Okay. So just putting that on. But going to rotate at high speed, and then I can slowly dig into the sample. You may notice that I haven't cleaned it yet. So it's really important to clean it before you use it, okay? So what I've done off camera that you didn't really see is that I cleaned the whole desk area here with bleach and then with alcohol, okay? So just making sure that this morning I was like standing on it, I was touching everything. So I made sure that bleach has been used to clean it. And then the same for my phone, which I've been using, with my bare hands. So I cleaned it with bleach and ethanol. A lot of ethanol to remove the bleach because I don't want it to rust. And that's the issue with bleach, of course, is that, all the metal bits, it's still shiny here. It's relatively new. But after a couple of years, it's going to be a bit rusty and everything, but, so be it. So yeah, this is pretty much it for now in terms of the equipment that I use and then the setup. I like to have a little bit of room. So, and not too many people around. And then, yeah, that's pretty much it. The other thing as well is there is a computer next to me or someone with a computer next to me who is going to take some notes, okay? Because, of course, what's important is to make sure that whatever samples arrive here on the surface, on this working surface, I know where it's coming from. So I have a specimen ID. I have some information about the specimen that the archaeologist is willing to share. It can be anything related to the location of the site, the country where it was found, dating, whether it is like a cultural period or an actual radiocarbon date already. Any information that could be used for my analysis. And then, of course, what is the sample? You know, what is a specimen? Is it like a complete skull coming from an articulated skeleton? On that skull, you know, how many teeth are left, how many have fallen after death, before death, and then pathological information maybe, like caries on the teeth or lesions on the ribs that may show some sign of tuberculosis. Anything that could be useful basically to have some information about the sample. And the date, you know, is it like a direct radiocarbon date, or is it like associated, a charcoal piece or another animal bone associated to the specimen? So anything that can be useful. It doesn't need to be collected during, you know, the day I'm sampling, because we're going to be in touch and then we can share information by email. But what's important is really the ID that you have already given to that specimen or to that sample, because I want to keep track of that. Okay, so there's a traceability that is needed here, because I'm going to give another unique ID in my lab. Okay, but that needs to be connected to your ID, so we can speak about the same thing later. And then the origin of the remains as you know, geographical origin. Yeah, so there's a table that Gluduk is filling on his computer at the moment. It has like a number of columns with information basically, and in lines I have every individual sample, and then we just, you know, fill that sample as we go. So the first column in that table for me is the internal laboratory ID, which is going to be ACAD 26058, for example, okay? And then the second column is going to be a makeshift ID that I'm going to give now, for example. So it's going to be like the 3rd of August, 2022, sample number 1. Okay, that's the first sample that I have today, and that's going to be the day my sampling ID basically, and then the archaeological ID, and then a bunch of other information. I don't know for you, but it's bloody hot in there. So this is it for setting up everything. It takes a little bit of time, but you know, as anything good, it always takes time to prepare. So what I'm going to do now is preparing like a sheet of alfoil that I'm going to use to cover the surface. And that one is going to stay here for quite a while, actually. It's really just to protect the desk, you know, I mean, because I don't want to do anything to it. So we're not really like in a space where we do this kind of work. So that's just a little bit of ethanol, so it sticks to the surface, it doesn't move anymore. Et voilà. Hop. Bleach. And so you notice as well that I put the bleach on the paper towel, not directly on the alfoil, because I prefer to be in control of the spraying, basically, instead of like spraying everywhere. Okay? And then ethanol. So ethanol is just like to remove the excess of bleach. And now I have like a very clean working surface, basically, where I'm confident that I can work on it and there's no contamination possible. So now my hands, you know, I mean, I have only one pair of gloves. I've touched a lot of things around, not necessarily super clean. What I'm going to do now is have a box of gloves next to me. And that's going to be my working gloves. Glove on glove is not necessarily easy to put on, so you will practice, I'm sure. And these gloves, now I can touch things, and then if I have even you know, suspicion that it's contaminated with human DNA, I remove it in the bin, change it, okay? So what you haven't seen before is that, you know, I cleaned this with bleach and ethanol, and then I cleaned this as well with bleach and ethanol. And the thing that I haven't cleaned yet is the actual bit, the Dremel bit. So if I need it, it's going to be cleaned. Ethanol, a lot of it, just to make sure that I remove all excess bleach. And then that can stay here. Okay, so the magic box. So I see there's already like something written on the box. So I'm going to ask the archaeologist what that information is. So brain, PR, Arkeologi, PS or P5, I don't know.
Yeah. PS, what's the dance spell?
Okay. And then sample, Tulang, and
Until.
So that's one sample.
okay. So is there information here that is actually related to the samples inside? Not really. It's just like general information. So I don't need to record that.
But there's another information just. Yes.
Aha. Situs megalithic, Swobu, Kota, Jayapura.
Let's just be
So that information is important. So this is something that we need to report in our notes. So situs megalithic,
Metode, Jayapura.
Swobu.
Ya, the name is nama situsnya Sobu, kotanya Jayapura, Papua. Okay.
So in this box, there is what looks like a temporal bone. And then 1, 2, 3, bags with small samples, and that looks like teeth. Is that correct? Yep. So let's start with teeth. So one by one. So I have different labels on those bags, so I assume that it's different individuals. Yep. Different individuals. Now, so I move that box away from where I work, okay, because maybe I'm going to do something to that sample and I don't want it to contaminate the other samples. I'm not going to do anything. So there's a lot of information recorded here, okay, that the archaeologist has provided with the sample. So we know that we can identify the sample whenever we work with it. And usually what I like to do is take a photo of that. Do you allow me to take a photo? Or I'm just taking a fake photo, basically. So, you know, I actually... Oh yeah, the reason why I'm wearing that as well is because of the bleach, okay? I don't want my clothes to be bleached as well. Okay. So why do I change my gloves now? Because I touched that bag, and probably that bag has been touched by bare hands, mine in particular yesterday, but other people as well. So if you're limited with your number of gloves, you know, I mean, maybe you brought just a handful and then you don't have enough, then you can clean the glove with bleach, okay? And then with ethanol. That's okay to do that, okay? It's just that at some point, the glove is going to be like a bit sticky and a bit, so you need to change it, but you can definitely clean it. So now I have a bag, I have my phone that I have cleaned before, I put the camera on, take a photo, and now I have a record, okay, that I'm going to keep. And I'm, at least, if I start writing information in my database and I do a mistake, I can go back to the photo and I know that I'm going to have the right information, okay? So on that label, there's a lot of information. There's information about, you know, where the sample is coming from, so the site, location, etc. There's a date as well of actual excavation. And then there's other information that I don't understand, but you understand. And so, you know, I would ask you, okay, what is useful here? But there's definitely a unique identification number, which is SRB/TL1A, et cetera, et cetera. So that is the archaeological number that we record in the database, basically, okay? And then from there, it is a tooth. So I want to know where that tooth comes from. Was it in the mandible, and then you took it and placed it in the bag? Or was it like somewhere on the ground, and you took it and placed it in the bag? So which one?
Kami tanyain dari mana tulang dari soket kan.
Kalau di situ baru sih bisa.
Oh iya.
Di keterangannya.
That's a,
Oh, it's written on it?
Bahasa Inggris juga.
Speed teraduk, speednya teraduk. Speed teraduk tengkorak.
Eskalasi.
Iya, kalau tengkorak aja sih. TL1A. Maxilla.
Maxilla. Okay. And so by the size of it, it looks like a molar. Do we know which molar? M3 on the right, left, right? Okay, so 1, 3. Okay. No, so that would be M3. Oh, so that would be 1, 8. Okay. So that's the wisdom molar, basically, on the right side, up. Yeah, up. So 1, 8. Okay. Anyway, as long as we understand which one it is, all good. And then at that point, you know, I mean, ideally what I would like to do with this sample is bring it to my lab. So leave it intact here, okay? And just like bring it to my lab. So in my lab, I can basically clean the roots, cut the roots, and do everything in an environment that is protected, basically. But if you want to have, you know, just the tooth back, then in that case, I need to take it, cut the root, and then bring it to my lab. And the root will be used for DNA extraction. But that means as well that if I do that, cutting the roots, we're going to open a hole and the pulp chamber is going to be exposed. So if one day you want to do pathological analysis, you know, like is there a special virus or bacteria in that individual, then that's going to create some problems. So my word of advice, my preferred way of doing things is to take the whole tooth, bring it to my lab, work on it. But first we agree, okay, so do I take the roots? Do I, you know, cut the tooth? Do I destroy it completely because you don't need it? Then we need to agree on that. And then once I'm in my lab, I'm like, oh, okay, so I remember we just can take one root and then we send it back. Okay, so this is always the same thing. Taking a sample into my lab doesn't mean that this sample is going to disappear forever. Okay, taking the sample to the lab just means I can work in the cleanest conditions possible and then place the tooth back into its bag and then collect all the samples, come back to you. Thanks very much. This is, you know, what's left of the remains, the samples. So just out of curiosity, I'm going to have a look at the sample. No, sorry, I'm just playing with you. Okay, so we have a tooth here. The enamel is, the structure of the enamel is good. Oh, sorry. So the structure of the enamel is good. There's no cracks. The color is good. It's shiny. It's looking pretty good. There is a little bit of dirt on the roots, like the gray area, if you can see it. And then I can see the tip of the root here is a little bit discolored. It's a little bit like chalk, you know, like a bit porous. So probably not the best place where to sample because I assume that this is probably a little bit poor in DNA. But that root here, you can see that it's got a yellowish, creamy kind of color, and it looks in a much better preservation state, basically, than the other one. So if I was to sample that in my lab, what I would do is take my Dremel tool, and I don't know if you just noticed, but I'm manipulating my samples with those 3 fingers on each end, and these other fingers have never touched anything, okay? So I can use them basically to manipulate something else, okay? So I mean, right now I'm handling my Dremel between my fingers that didn't touch the samples. So these are things, that you learn. Same, if I need to move and open a handle, I use my pinky finger and I make sure that I minimize contamination. So in this sample, in this case, if we were working together, I would ask you, can I take that sample to Australia, to my lab? I'm going to cut the root on this side here. And then, depending on what you want to do later, I can drill inside the pulp chamber just a little bit to collect bone dentin powder, and that we can put it aside maybe for pathological or pathogen analysis next year, in 2 years. It's less of a priority than looking at the human DNA for now. Or you tell me, no, it is a priority because I have identified that this individual looks like it's sick. It has tuberculosis, for example. I would like to know if I can detect or Yersinia pestis or anything, I would like to know if there is something. And in that case, sample the inside, use it for DNA extraction, and then we go forward. And then once I have cut the root, then we can, take the root, place it in a sample bag, place it in one of those sample bags, where I'm going to copy the exact same label as this one, okay? So I know it's coming from the same place, plus a sample ID. And then put that back into the bag, the original bag, and then send it back to you. How does that work actually in Indonesia? Is it possible to use FedEx, for example, and send stuff back to the country? Or do I need to come personally, or Gluduk or Leonard, and then carry it and bring it back? How does that work? There's permits for that?
Yeah, I think it's easier to carry the sample because we don't start with FedEx. Something might be happened.
Okay.
During the process.
Okay.
So it might be lost. Yeah, except if we bring ourselves.
Okay. So yeah, and if I were to take that sample to my lab, so I would not take that, the aluminum foil, because you probably touched it with your fingers. So I would just remove it, place it in a new bag, and then give that back to you. So you have a record, it's empty, but you know that there was a sample in there, and on it, I write sampled by Bastien, 3rd of August, 2022, and you know what happened to that sample. And then when you need it, you call me and say, hey, I want it. So yeah, so that's what I would do for a tooth. And I'm still unsure if we actually sample it or not.
Oh. Oh.
Jadi memang kan batang ini akan kita kerjakan nantinya. Nanti di PKS sama Pak Irfan itu kan Prof juga masuk. Mungkin kalau memang sampel, if you think the sample is good enough,
yeah,
like the temporal bone, maka boleh kalau misalnya kita kita beneran sampling, tapi nanti disimpan ini dulu gitu. Nah, kalau misalnya memang dari kemarin boleh, jadi kita bisa langsung praktek dan sekalian nanti untuk penelitian juga gitu. Mumpung lagi ada yang memang bisa mengajarkan dan ahlinya datang gitu.
So, kalau sampel saja itu ya boleh lah, ya kan? Tadi bicara kita kan. Kalau itu yang lain yang serius,
Yang
Kita pasti dapat hasil yang terharapkan.
kalau dari sebenarnya kalau dari Gigi sih tidak akan diapa-apakan, paling nggak bakal we do not need to drill the tooth, right?
Sorry?
We do not need to drill the tooth or cut the tooth right now.
Not now. No.
Gigi nggak ada. before the temporal.
Yes. What I want to know is, do I put this tooth back in a new bag? And then we put it aside to bring it to Australia. Or just I leave it like this, and that's it. Or you can leave it like this.
Yeah,
And that's later. Or you can leave it like this.
yeah,
And that's later.
Nah, di sini kan.
Or you can leave it like this.
yeah,
And that's later.
Nah, di sini kan.
Oh, you-
Bahan kurang.
So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples. Just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples, just to see, how they look like. So I'm just going to look at the other samples. If we scratch the surface and remove this dirt, I'm pretty sure that underneath the bone is going to be relatively good. It has some weight to it. Okay, so it is relatively well preserved, and I don't see any cracks or any crumbliness on the roots. I mean, the roots are looking relatively healthy, not discolored. It's just covered with dirt, basically. So it looks like a good sample as well. And when I say a good or a bad sample, by the way, I don't guarantee there is DNA or not, okay? It's just that there's a chance that we can have some DNA in there. Good one. Okay. Next gift. ST1, S18, speed 4. So here, Oh yeah, and of course, I mean, so this tooth is a molar, but we can't really know. I mean, it's possible maybe with the root system to know where it comes from, but it's going to be hard to identify where it was, the previous one, the one before. We don't know what tooth it is. Ta-da! Okay, what do we have here? So we have another molar, a premolar maybe, actually. Very worn, so the top of the crown is completely worn out. Quite interesting, actually. So again, that's probably a sample that was exposed to water. Very light. I mean, so there's not much left in that sample. The roots, so the tips of the two roots has been broken. So it was probably like very crumbly, very fragile, and it just like fell off. This one is probably also found underground, or was it removed from the grave, but not in the mandible or not in the maxilla? Yeah. So this one is a tough one. I don't know what to say about it. I don't know if it's going to be good for DNA or not. It looks like relatively damaged, but I will need to remove the surface and see how it is inside to say, okay, I can actually, you know, hope for DNA. But I've seen, you know, teeth in a much worse shape giving DNA. So it is completely possible that it gives something. At least the rest of the root now has a nice structure. It's not breaking. It's not like, you know, very fragile. So I'm confident that at least there's a little bit of good preservation at the structural level. Is there any DNA in there? We can test it, but I can't really say anything. So in this kind of situation, this one and the previous one, they were found outside of the socket, outside of the mandible or the maxilla. So that means that I do need in the lab to scrape the surface, so remove like the outer layer before I cut. So I remove the dirt, I remove anything that has been at the surface that does not contain human DNA, basically, or maybe that contains your DNA. And then only after that I can eventually cut it. So the idea of, you know, putting it into a solution and then just digesting the surface, that's not a good idea with this one, because it's possible that there's too much contaminant DNA, and then we will lose the actual human DNA in there. So I would say it's possible to try it, but there's no guarantee that it's going to work. That's a limit kind of a scenario, okay? And again, I mean, so 1 tooth roots, and they're relatively big here, so that would probably provide, after removing the surface, that would probably provide like maybe 70, 100 milligrams of powder for one. So that's enough for DNA, basically. So it's a good sample for that. Very, very impressive pattern here. Are people actually chewing ropes or vegetal fibers or anything like this, like making ropes? Because this is really impressive. It's really like eating.
Sesuatu yang berserat. Probably, but
yeah.
Sigurd, no idea what happens.
Yeah.
Actually, yeah. Or maybe just another old individual.
Yeah, even an old individual, I mean, the top of the crown is really like gone, basically. So that would be someone who has, I don't know, eaten a lot of grit, maybe, or like who was weaving shells. Okay, okay.
Yang tampak di tangan itu banyak ini terang. Buat kemudian
Okay. So yeah, thanks very much for sharing this, because we have 3 different examples of preservation, basically. And so that's really useful, because now you have an idea of, the confidence we can have about, yes, the first tooth was good, probably contains DNA. We can definitely work with it. It was taken from the maxillary, so relatively free of contaminant DNA. So that's good sample. The second one, covered with dirt, so we need to remove the surface. One root was looking not so good, the other one was looking really good. So there's probably DNA as well. And the third one, found outside of the socket as well, in the ground, need to remove the surface. It looks a bit less well preserved, but maybe there's DNA in there. So you don't have like the example where as soon as I touch it, it's powder. So that, forget about it. It's not good at all. Now, temporal bone. So in this magic box, we have another sample that is not a tooth. And so my first question is, that temporal bone, was it detached from the skull in the grave, or did it fell off after you started to excavate?
During the excavation.
During the excavation.
They dig cell or and find grave there, secondary grave.
Mm-hmm.
After that, they say to us to go there or find the skeleton. If I find skeleton, but before people even before us, and we continue to find it not only cranium, we find the skeleton still.
Okay.
And this one, I take one of the cranium.
Okay. So it was in situ, it was attached to the cranium, and it's just when taking the cranium, the temporal bone just like fell. Yep. That's a good looking one. So here we go. Zoom in. Yeah. Oh, good. Yeah, that's good. So a nice temporal bone here, definitely heavy. So there is, organic material that is preserved in this piece of bone. And there's a little bit of sand and dirt, falling as I'm moving it around, which is okay. So that's the left temporal bone. We can see the ear canal here. So that hole there. And that's the petrous pyramid inside. And then basically on that side here, we have like the jugular foramen. And then the maxillary foramen is here, but it's yeah, it's just like broken. So basically the part of that bone that I'm interested in is it's below that part here. So that's where the actually, sorry. No. Yes, so that's where the jugular should be. So where I should, drill, basically, it's below that area here. So I can remove that bridge here and then remove the outside of the bone. And then at some point, I'm going to hit the region where the cochlea is, basically. And then we can start like drilling inside. And then the powder is going to fall through the ear canal. So drilling here, basically, and then the powder coming off the other side on this side of the temporal bone. So yeah, that's a good one. I'm just removing the dirt from the ear. Do I go and? Sorry? Yeah. And so in this case, this is so either you have like the whole skull, or you have like a fragment, or you have just the petrosal part detached from the bone. So again, this is a situation where I would prefer to have that in my lab and then process that in my lab in the cleanest environment possible. But this is also something that you can do here, and just like making sure that you clean the surface. I put like another, I'm going to do it for fake, okay? That's just not for, so now I have my sample. I think, okay, I can do something with it. So here, there's a little bit of dirt. It doesn't really matter, because what I'm going to do now is prepare for sampling. So new gloves. So brand new piece of ice foil. I'm going to use that basically to collect the powder in it. And then when I'm ready to collect the powder, I just hold it, take a tube, and then put the powder in it. Okay? But before then, what do? I clean. bleach. Ethanol. Let it dry a bit. In the meantime, I prepare a tube. So I prepare a tube here with a label. So ideally, I want the same label as on the bag. Okay. And then the aircon is making my paper fly. And then I have my tube ready, okay, for collection of sample. Then I can bring my sample in. So this is ready for the collection of the clean bone powder inside the petrosal bone. But first, like I said earlier, I want to remove the surface, like this ridge, that is hiding the cochlea area. So I'm going to put that on the side, basically for now, and then work here, take my drill, and then gently. So this is how it goes. Gently start drilling in that area here. So remove the bone surface until I start seeing two small holes, and that's basically the bony labyrinth appearing. And then I put that aside. That's the bone we're not going to use. And then I bring my clean sheet of alfoil, and now I'm going to make sure that the ear canal is facing down, and I'm going to keep drilling inside, slowly going around. So I collect, basically I make the bone inside a powder, okay? And that's going to fall through the ear canal. And then there's going to be like a hole, basically a small hole in the sample, and powder here. And then I make sure that there's enough powder basically to probably go to, yeah, that going to that level. I mean, a bit more is good, but you see that you don't need much, okay? So just like the tip of the tube, that should be enough. So that's how we do it, basically. And then you make sure that the label is correct, your name, the date of the sampling, and then you bleach the tube. Bleach the tube, put it in a clean bag, done. And that can go to the lab, basically, okay? And yeah, that's about it. I mean, do you have any questions?
I've got a question from this here. How about if the sample is found underwater or maybe seawater? How do you clean the sample? Because it's already wet.
Yes, yes. So basically, the idea is to try and dry it. And drying it fast is not a good idea, because usually that's going to create some cracks and degrade the sample. So what I would do is either, so take it from the water, wrap it in paper, absorbing paper, remove most of the water that is on the outside of the bone. And then once that is done, and you can maybe, you know, like do it on the spot and then wrap it overnight. And then once that is done, either you have some silica powder that you can, you know, place in a box and then put your sample in a plastic, open plastic bag inside that box, and then let the silica absorb the humidity. Or you keep it, you know, wrapped in some clean paper on the surface, and then you wait maybe days, maybe weeks before it's actually dry. Alfoil is going to retain the humidity to trap it inside. So yeah, or you leave it like open, but you know, I would rather like have it wrapped in paper for quite a while, and then what you can do is leave it in a plastic bag that is open, you know, so at least it's kind of protected, and let the temperature do its job, basically. Yeah. So I changed my gloves just to wrap that up again. And so again, you know, I mean, I would be more confident to do this work in the lab, if possible. So send the whole thing or bring it, you know, to the lab and then do it there. But if you want to keep that sample, then yeah, maybe you can collect the powder yourself and then send it to me. But I would prefer to collect the powder myself, basically, in my clean lab. Cool. Thanks very much for bringing those. So we could do like a little bit of a demonstration. Is there any other question? Do you have anything that was not clear or?
Ini kan sering kali kalau kita ke lapangan dan dapat temuan yang seperti ini, kondisi tulang kan kadang para contohnya ini yang dari kemarin kita sudah pengalaman kita ditunggunya biar tulangnya tidak hancur bisa pakai konsolidan aseton dan paranoid itu sebagai pengganti lem. Memang tidak disarankan untuk menggunakan lem pada saat itu, tapi katanya kalau menggunakan aseton dan paranoid ini campur di mix dengan komposisi yang masih mobil, yang jelas itu adalah konsolidan di mix untuk dioleskan di atas permukaan tulang. Itu biasanya akan menguatkan tulang. Biasanya masih pada materinya, masih pada masih pada panahnya. Nah, itu bisa katanya dilakukan, bisa kita lakukan sampai proses penanganan pemindahan pengangkatan. Jadi kita biasanya di box, kalau kita tidak kemudian satu, dan kita angkat matriksnya nanti dipreparasi di atas meja lab kerja lagi. Ya, buat dibersihin satu-satu lagi di atas meja ya screen. Jadi kalau untuk misalnya modelnya steril seperti ini memang butuh meja kerja yang lain lagi untuk setelah dibersihkan seperti itu. Nah, efektif sih efektif menurut kami karena itu memudahkan kami mengangkut semuanya karena sudah solid, sudah keras, itu sudah tidak rapuh lagi. Nah, itu bagaimana itu kira-kira?
Tadi itunya apa namanya? Nyawanya?
Aseton.
Sepan.
Pak aseton dan Pak B itu 273.
So,
Mas Glodok, I'm the moderator, so I can translate. Yeah. The stage is ours.
I'm only listening to you and everything.
Yeah. So the question from Pak Fahri. Suara masih masuk ke Zoom? Okay. The problem is we always find fragile bones in the field, and Fahri mentioned that to protect and to make the bone stronger before we took it from the field, we use the Polaroid and
Paraloid and aseton.
Paraloid and aceton. So the question is it possible to have the DNA preserved when we apply those chemical composition?
Well, acetone is going to create some issues when we try to manipulate the DNA. So enzymes and everything are going to be sensitive to that. Plus, I'm not too sure what it does to the DNA. I don't know, actually. I don't know what it does to the DNA, but definitely presence of chemicals. When we start extracting the DNA and trying to amplify it and sequence it, that's going to maybe create some issues. So I would avoid, yeah.
Ya, ini sih Mas Niko, itu bisa kita lakukan karena nanti pada saat preparasi di meja laboratorium nantinya itu akan ada perlakuan khusus yang bisa untuk menghilangkan kandungan konsolidannya itu.
Iya, tapi mungkin kita tidak bisa mengontrol apa yang sudah masuk ke dalamnya itu untuk DNA, mungkin itu. Related to the question, is it possible like we like based on his experience, is it possible like we took the small pieces of the bone for the DNA analysis while we protect the whole, the big bone to be preserved? So I think that is the win-win solution.
Exactly.
For everyone, yeah.
Exactly.
It depends on the goals of the research.
Temporal and
you can
maybe.
you can take the best piece for
So
the DNA analysis.
exactly. So of course you're completely right. I mean the targets, for sampling for ancient DNA are probably the teeth, the temporal bone, maybe the phalanx, and the rest of the skeleton, I would say, is it is safe to prepare it the way you want to preserve it as much as as long as you can. So yeah.
Thank you, Pak. Thank you, Pak. Lebih aman, jangan pakai buat yang mau dijadikan sampel DNA, jangan pakai kandungan kimia.
Sama.
Nyambung dari pertanyaan tadi Pak Riz. Kita menemukan tulang sebaiknya perlakuannya gunakan apa? Kali-kali kita melihat bahwa untuk bersihkan tulang di dalam aplikasi itu kan menggunakan air atau alkohol kan? Ya kayak kotor gitu kan? Melihat tulang apa gitu kan?
Oh, okay. So his question is, when we found a bone, we are really not sure whether it is a human or animals or faunal bones.
Oh yeah.
And usually we use water or any liquid to identify it.
Yeah.
So is it? Yeah. The question is, how is the ideal works to identify the bone, whereas we don't know exactly what is it?
Yeah. Tricky question. It's a tricky question. I would say don't use water in any circumstance. So best maybe would be like to brush the bone as much as possible, and then try and see, if you have the possibility to identify the bone like this. If really you need to use water, I guess it's because it's a fragment that is too small, or maybe, okay, it's an isolated tooth, and then you need to know, okay, it's a human or a dog or whatever, I don't know. Yeah, the use of water is no good, because then after that you need to dry the sample, and or maybe it's already wet. If it's already wet, then you can use water, but the water you use might actually contain bacteria that were not in the ground, and so you're going to add a layer of contaminants possibly. It's a tough question, and I don't think, again, it is not wrong to clean the bone and try and identify it, or the tooth, okay? It's just keep a record of it. Keep a record, say, hey, this particular sample, I was unable to identify it, so I had to use a bit of water to rinse it and to remove the dirt, and you write it in your notes, and when we have the DNA results, then we can say, oh, it's funny that, we have these particular bacteria, for example, because it's not part of the environment, when it's coming from somewhere else. And then we go back to your notes and like, oh, water was used. And, but if the sample was dry and you put water on it, then I would say, no, don't do it. Use water only if it's already wet, because you don't want to add like water on the dry sample and then risk to damage it or contaminate it. Does that make sense? Yeah.
Yeah, sure. Yeah. In mostly in prehistoric excavation, we use the bamboo stick to dig. So is it possible, like the contamination from the bamboo stick when it touch directly to the bones, or it's fine?
Yeah, I think it's fine. I mean, there's going to be a little bit of, transfer of the bamboo DNA maybe to the surface of the bone, but at that point, I don't think it's going to be like you oily fingers, where the DNA sticks really to the. So it's interesting still to know that bamboo was used, because, for example, I don't know, we do a study on the diet of those people, and we use like a little bit of dental calculus, and then we find out that there's bamboo in it. Maybe it's just like a fragment of bamboo that was trapped in there, and now that we know that you use bamboo, then we're like a bit suspicious, it's something that is not expected in the diet of people. And why do we observe bamboo now? It's because there was a tool that was used to excavate. So
yeah. Got it.
Just, yeah. And like I said earlier, it's really like an investigation that we do. So I don't think there is any wrong way of doing things, It's just that as long as we keep a record and we are, not forgetting or hiding things, then when we look at the data, we can say, hey, something's weird, but I have an explanation for it. It's because of this or that.
Ada pertanyaan lagi dari ruangan advisional mungkin? Mbak, agak kencang, maaf. Agak kencang.
Membersihkan tangan itu dengan luas. Umpamanya ada tulang di sebelah kanan, kemudian tulang di sebelah kiri. Apakah boleh kuas yang di sini dipakai ke sini juga atau satu-satu?
Yeah, Professor Llamas, it's a very simple question. Can we use a brush to clean multiple bones, or we need only one brush, specific brush for each bones?
Yeah, that's a tricky one. I mean, it's impossible to have one brush to clean one bone. Okay, it's impossible. So in this case, it's really a matter of removing as much dirt with the one brush, and then I don't know. I don't know what to do here. For sure, you're going to reuse the same brush to brush multiple specimens. Whether that's going to be a contaminating issue. Wow, a simple question, but a very difficult answer.
What if we use bleach maybe to clean, and then we use it after? Like
you could use bleach on the brush, but the problem is then you need to wait until the brush is dry again. So it is a difficult question. I don't know the practicality of it basically, and this is why most of the time I prefer people to leave the dirt on the sample. And then in the lab, usually what I do is that I clean myself the dirt, basically, using, the Dremel tool or something. And yeah, it limits the risk of contamination. But just the mechanical action of brushing and then going to another sample, if the brush is a soft one, I don't think it's going to be a problem. If it's a bit of a hard brush, like a toothbrush or something, then there's a chance that you may actually have a bit of dust, bone dust, bone powder, if you really go too far and the bone is a little bit like brittle, and then you transfer it onto the next one. Yeah, I don't have a clear answer, I'm sorry. And to be fair, but this is why I love this question. It's because common sense, you have a situation and you need to find the best way, to minimize risk. And sometimes there's no best way. There's one way, or maybe another one, or maybe a third way to deal with the situation. And well, you pick one, and then you just do it. And again, you take a note and just say, so I have this grave and I have 6 specimens and I have only one brush. I use the same brush for the 6 specimens. Boom, it's in your notes. We will know about it later on. And then maybe that will help us understanding some issues in the data. Or maybe there's no issues in the data because actually we can brush multiple individuals and there's no risk. I don't know. I really don't know. But, if you come to me and say, be careful because, on that day, I used the one brush to clean like 5 samples, then I'll be, okay, cool. So the risk is that you may have transferred some surface, contaminants to another bone. So what do? In the lab, I use my disc, and then I remove the surface. And then once the surface is gone, I'm pretty confident that inside, it's okay. The brush never reached it, so I can deal with that. So, that's a way around it. Because otherwise, logistically, to bring like 60 brushes, to the side, it's just crazy. So it is not possible. So, but, you told me I use only one brush, be careful with that. And then I'll be like, okay, so do we agree, that I can like remove the surface, clean the bone before I sample it? And you tell me, yeah, if that's the best solution, then let's do that. And so I'll do it.
Yeah. Thank you. It was a long answer for a short question.
I'm sorry. I'm sorry.
No problem, no problem.
I had to think about this one.
Really great. So, baik, jika tidak ada lagi pertanyaan, mungkin akan saya tutup sesi untuk hari ini, kegiatan hari ini. Thank you so much, Professor Llamas, for coming and for sharing everything you know about DNA analysis.
You're welcome. Hopefully it's going to be useful to you guys. And I'm really thank you very much for the questions and for the interaction. I really enjoyed it. So it's good.
Ya, terima kasih juga untuk teman-teman dari BRIN yang berada di sebelah kiri saya sudah mendampingi di sini, dan Bapak Ibu sekalian yang hadir di ruangan RP Siono dan di ruangan Zoom. Mohon maaf jika masih banyak kekurangan dalam terselenggaranya acara ini. Dengan demikian saya tutup sesi sore dan saya akan kembalikan kepada MC.
Dan terima kasih untuk informasi-informasi yang telah diberikan tadi dari Mr. Bastien Llamas. Thank you very much. And Mr. Niko Alamsyah, also thank you very much. Sebelum ditutup, mungkin akan ada sedikit kata-kata penutup dari Kepala Pusat Sejarah dan Prasejarah, Bapak Irfan Mahmud. Oleh karena itu, saya serahkan terlebih dahulu kepada Pak Irfan. Silakan Pak Irfan.
Ya, selamat sore, good evening. Thank you, Mr. Bastien. Presentation good. Pak Gluduk, ini Pak Gluduk bikin takut Pak Heri namanya. Selalu kalau disebut dia terkejut. Ya terima kasih Pak Mas Gluduk, Pak Leo, Bu Hera mungkin yang ada di Zoom. Terima kasih Bu. Ini saya kira materi yang sangat memperkaya arkeologi Indonesia ya. Sesuatu yang lama sekali kita tinggal. Mungkin dalam satu dasawarsa sudah ada perhatian seperti Mas Niko sudah, lalu ada beberapa lah yang sudah memberi perhatian. Harapan kita ada dari kami juga nanti ya, lebih memperdalam lagi di mengikuti Mas Gluduk dan Mas Leo nanti. Mudah-mudahan ada ya kesempatan dari sivitas kami di ini nanti lebih memperdalam hal yang sama, sehingga kerjasama kita merupakan kerjasama yang lebih panjang lagi dan bisa menghasilkan dan mengisi kekosongan informasi tentang sejarah leluhur kita di masa lalu. Bukan saja garis historis migrasi mereka, tetapi mungkin saja ada hal-hal yang seperti sering didiskusikan mengenai penyakit-penyakit endemik yang mungkin ada tersimpan di dalam DNA kita, lalu kebiasaan-kebiasaan, pola-pola yang berpengaruh, pola hidup yang berpengaruh yang mungkin terekam dalam DNA yang di dalam artefak bisa saja menjadi asumsi-asumsi dan kegelisahan untuk menjawabnya bisa dibantu oleh DNA. Saya kira memang pengetahuan ini sangat penting buat kami ya, dan kerjasama ini tentu menjembatani lahirnya satu informasi yang lebih banyak lagi, karena paling tidak di dalam studi-studi kami tidak banyak yang bisa menjelaskan itu dalam kaitan dengan artefak. Nah ini mohon bantuannya dan kerjasamanya, dan kami juga mengapresiasi ya bahwa apa yang akan kita lakukan ke depan dengan segera kami akan bahu-membahu lah untuk lebih memperlancar lah apa yang kita butuhkan sama-sama. Tentu banyak kekurangan mungkin ya kawan-kawan semua yang hadir maupun di Zoom maupun di secara luring, mohon maaf karena tentu dalam banyak hal kita tidak bisa memperkirakan banyak hal yang mungkin terjadi. Kami juga berterima kasih Mas Gluduk, Pak Leo, teman-teman di tim webinar ini sudah membantu kita, Mbak Indah, siapa lagi, Mas Andri yang di belakang layar tidak ada di sini, tetapi mereka tetap bekerja untuk kita dalam mempublikasikan secara langsung apa yang terjadi di sini begitu. Saya juga menitip kawan-kawan di Makassar dan Papua untuk untuk sama-sama ya saling berkolaborasi mempersiapkan apa yang bisa mempertautkan kerjasama kita, dan tentu saja bukan cuma data-data sampel tulang dan giginya, tapi juga mungkin kita mempersiapkan banyak hal yang terkait dengan peran-peran arkeologi atau isu-isu arkeologi yang bisa mengisi penjelasan-penjelasan yang setara begitu. Di sini juga tentu saya berterima kasih secara khusus ya kawan-kawan yang di sini beberapa hari yang lalu sudah mempersiapkan, ada Mas Wewe, Riady, kemudian Mas Niko dan lain-lain ya yang saya tidak bisa sebutkan satu-satu. Tidak kurang rasa terima kasih saya ya sebagai pimpinan di Pusat Riset, tanpa melihat batas-batas kelembagaan, kami tetap sharing dan berharap ini merupakan kolaborasi yang baik untuk kita semua. Saya kira itu, terima kasih semuanya atas ilmunya begitu. Terima kasih kehadirannya Bapak Ibu yang secara langsung di sini luring dan yang daring. Mudah-mudahan ini akan ada berikutnya ya Mas Leo ya. Kapan-kapan lagi kita akan mudah-mudahan ada hasil-hasil yang baik dan kita bisa publish secara baik. Terima kasih semuanya. Sekali lagi mohon maaf jika ada yang